Great. Thank you much. Bye. Hello, everyone. You guys all got quiet. What is this? Welcome to BB 450. My name is Kevin Ahern, and I will be your instructor du jour. You guys have probably heard that biochemistry is hell, and after sitting in here and sweating, you know it is hell, right? There's no question. In fact, people who know it's hell are sitting up there, I think. Is it warm up there? Yeah, well, hot air rises, you know. So happy to have you guys here. I look forward to a good term. I hope you had a good summer. What I'm going to do today is go through a bit of just sort of logistics and introduce some TAs to you, at least the TAs who were able to make it to class today, and engage even a little bit of dialogue. So that's kind of cool. Let me start with the TAs first. So can I ask my TAs to stand up so I can point them out? Okay, Nick, right here. There's Sarah Cotting. This is Rakan Kaki, and this is Ariam. And there's one other TA, and that is Sarah Hill Skinner. She's unable to be here because she has class at this time. But I just wanted to introduce them to you. They will be leading recitations for you, and I'll be posting on the web soon their names, contact information, which recitations they're doing, and all that sort of stuff. So the first question that you probably have is, do we have to go to recitation this week? And the answer is yes, you have to go to recitation this week. Okay, so, and they'll be there taking attendance. So recitation is something that you need to attend. And if you don't attend recitation, then you will lose points, etc. So make sure that you attend recitation. And I'm going to trip on that if I'm not careful. So recitations are there for you. They are problem-solving sessions. And I tell the TAs that they are obviously to take attendance. And the first thing that they're supposed to do is they're supposed to ask, do you have any questions? All right? So if you have questions, or if you go to recitation, take that thing off. Okay? If you go to recitation, you should be prepared with questions because that's their time to help you with that. They're all set up. They've had, either in some cases have had this course, or in other cases have had other courses that are equivalent where they can help you. So make sure you go with questions because I think you'll have a much better experience if you do that. One of the other first questions I get from students is, well, I'm going to need a tutor. And I'm also happy to help you get a tutor. I've got plenty of very good students in biochemistry who can tutor. They charge, all right? So TAs are free tutors during their office hours. So if you want to save money, that's a good place to start. Check out their office hours. You do not have to go to the TA's office hours for the recitation that you're in. So if you're in Nick's section and you can't make his office hours, but you can make Sarah's office hours, that's perfectly okay to do. Okay? So, clear as mud on that? You guys look tired already, man. The term is just getting started. Okay. So don't want you to get too tired. So that's the sort of logistics of recitations. I keep a lot of office hours. In fact, when you look online, for me, you will see that my office hours, my calendar is TBA. But you can always see my calendar by looking right here. Okay? So if you click on that, you can see my calendar, where I am, what I'm doing, etc. And whenever I don't have, I'm a busy guy. Whenever I don't have something else going on, I'm available. So please come by. You don't have to make an appointment. All right? And I'm more than happy to meet with you. So one of the things I want to do is hopefully help you to address your anxieties over biochemistry. How many people have anxieties over biochemistry? Oh, yeah. You know, when I took biochemistry for the first time, I had anxieties and I hated it. I really hated biochemistry the first time I took it. Okay? I had somebody who had a real badass attitude. And I wasn't a biochemistry major as an undergrad. I was a total goofball, even more than I am today. Okay? I didn't get into biochemistry until later. And it was partly because I didn't really understand, okay, that there were all the cool things that were in it. So one of the things I hope to communicate to you are all the cool things that are in it. And I really want to cut through that anxiety that you have about a subject that I know is important for you. Many of you in here are pre-med, pre-dental, pre-pharmacy. Virtually everybody is in here because they have to take it. Not too many in here because they want to take it. Right? I understand that. All right? So I want you to buy into this. I want you to enjoy it. I'm not going to try to make a biochemistry major out of you. That's not my job. But my job is to show you the molecular basis of life, okay, and communicate to you the value and importance of that. And if I can do that, okay, then I will have done my job. I'll feel very good about that. So I want you to have this enthusiasm for this subject. I want you to get in and get your hands dirty, I guess. I don't know what the expression is, but I want you to dig into it and learn from this. Okay, so that's a very important thing. Now, this is a big class. Gesundheit, you're allergic to biochemistry. You're probably not alone. Probably the whole class will be sneezing before you know it. So, or itching, or or something like that. Break out in a rash. As I said, I try to be as accessible as I can. And if you're having difficulty getting a hold of me or whatever, feel free to send me an email. You can give me a phone call. I'll give you my cell phone number. You can call me. Okay, I have no problem with you doing that. So you can get a hold of me any way that you want. You want my cell phone number? Write it down. 541. There's your first assignment. 908-4465. You can call me any time of the day or night. I turn my phone off at night. You won't bug me if you call me at night. I won't talk to you, but you won't. Okay? Feel free. Call me. All right? You have a question. You have a problem. That's what I'm here to do. Now, I want to be accessible to you. I want to be available to you. I videotape all my lectures. Okay? Videotape all my lectures. There's a camera going back there right now. I will post on the class webpage, which is the main page, which is not this page, obviously. I will post on the class webpage, if you click right here, the tentative schedule, you can see everything that's available in the course. You can see the outline, which I'll go through today. You can pull down all the figures. I'll post highlights after class describing what I thought were the highlights of what I talked about. There'll be streaming videos, streaming audios. You can download them. You can do whatever you want to with them. You can throw rocks at them. Though I wouldn't recommend doing that to your computer monitor. Okay? So I tend to get those posted within about 24 hours of giving a lecture. And so I think it's obviously important that that be up there in a timely fashion. And I try to do that. I can tell you, I can't assure you in every case the video will work. Okay? Usually once or twice a term, there's some technical problem, and either the video or the audio or both are gone. Okay? So I can't promise you it's going to be there. I can also tell you from my own experience, and I have tracked this, that those who come to class do better. And I think it's partly because people who don't come to class try to kid themselves into certain things. Oh, I'll watch the video, I'll do this, I'll do that. And that turns into a habit. And they don't watch the video, or they don't get that, or perhaps they couldn't ask a question because it's kind of hard to ask a question to a video. All right? And people who come to class do better. All right? My videos are there not to keep you from coming to class. My videos are there to provide a supplement to what I'm talking about here. I talk fast. I walk fast. In fact, if you want to see something really funny, download the video, run it fast, and watch how many times I go back and forth like a tennis player. Okay? It looks really comical because I'm a walker and I'm a talker. All right? So the videos are there to help you to get on top of the material. I'm going to go through a lot of material this term. My aim is not to give you the videos, or, oh, now I can say anything I want to. I can dump all this stuff on them, et cetera, et cetera. Okay? There's a lot of material, but I give you the videos so that you have another way to access that information. Maybe English isn't your first language. Maybe you need more time to sit and think and listen as that's going on. Okay? So those videos are there for that purpose. Okay. Clear as mud? All right. The syllabuses are, the syllabus is required reading, so download the syllabus and read it. You can see it. You can download it, in fact, right at the very top there where it says download here. Okay? Download the syllabus and read it. There will be a question on the first exam that will come straight from the syllabus. Okay? So make sure that you do that. And that sets the policies and blah, blah, blah for the course. As the TAs will tell you in the recitations, you are to go to the recitation that you're signed up for. If you decide to go from Nick's recitation to Sarah's recitation and you don't make arrangements with both of them, you will get no credit for attending. Okay? So you have to make arrangements if you swap, and they are not obligated to say yes. Okay? It causes an imposition for them. So if they can do it and they say yes, then that's fine. I have no problem with that. You don't need to ask me about that. You ask them about that. But if they say yes, great, they'll take care of it. If they say no, then you'll have to find somebody else or make some other arrangement, okay? Because their time is valuable too. So it's important to do that. So as I said, I don't get involved generally with recitations and so forth. Okay, that's a quick and dirty. Let's just open the floor. Let's talk a little bit about biochemistry. When I say biochemistry, tell me some of the worst things that you've heard about biochemistry. Listen, you can even include me if you want to. I don't care. But biochemistry in general. Yeah, what's the worst thing you've heard about biochemistry?
Lots of pathways to memorize.
Lots of pathways to memorize. Yeah, there are a lot of pathways to memorize. Yeah.
Memorize all 20 amino acids.
You don't have to memorize all 20 amino acids. At least not the structures of them. So, well, you should know the 20 amino acids. But if you're taking the majors course, you memorize structures. In this class, you won't memorize structures. All right? And one of the reasons I like to have this dialogue is I found last year when I was teaching the class, there were all kinds of urban legends that were going through the class. And I finally had to intervene because students were getting really freaked out. Okay, I'll tell you one that was going through. Ahern's averages are really low. Okay? Averages in the 40s, that's pretty common. Okay? That was going all the way through the class. This is before the first exam. And of course, the more you hear this sort of crap, the more anxious you get because you're convinced that I'm going to flunk the class, right? And as I told the class, first of all, I grade on the curve. But in the second place, in the 12-plus years I've been teaching, actually, I'm sorry, 15 years I've been, time flies. 15 years I've been teaching biochemistry. I've given exactly 3 exams where the average was lower than 60. Okay? But when I told that to the class, it was amazing the transformation that happened. Oh! Okay? Urban legends really are bad. Okay? What else have people heard about that's bad about biochemistry? Some of them are true. I don't know. What's bad about biochemistry? 20 amino acids. He makes us talk on the first day. Is that bad? He tells bad jokes. Memorizing pathways. You know, I'll be honest with you, memorizing pathways, I try to limit memorization as much as I can. And one of the things you're going to see is you're going to see an awful lot of molecules. There's only going to be a handful of structures you're going to memorize. A handful. And I'll tell you which ones. So there's enough things to know. I would much rather have you learn something than memorize something. I'll tell you something I will do on the first exam. I will give you every equation that you'll need. You will not have to memorize an equation. You will not have to memorize a structure on the first exam. Okay? I don't think you'll have any structures that memorize, at least specific structures, until the second exam. And that's maybe 4 or 5. What else? How about glycolysis? Okay, so glycolysis we'll cover partly on the second exam, partly on the final exam. And we'll go through that at that time. So I'll tell you at that time what specific things I think you should memorize. There are a few structures there. I don't make you memorize all 10 structures. There's 10 different structures in glycolysis. And the only structures in this class that I make you memorize are structures that I think will be important for you in another class. Okay? Structures that will be important in another class. So, for example, one of the structures that you're going to have to memorize in this class is glucose. Glucose is the most common sugar on the face of the earth. Okay? You won't take a class at any kind of molecular level where you won't have to know glucose. So I think, okay, I'm doing you a favor by making you memorize glucose because once you've learned it once, learning it the second time makes a lot more sense. Am I going to make you learn 2-phosphoglycerate? Well, probably not. You're probably not going to use that again, right? So I do try to emphasize things that I think will be important for you and things that you will do later. My aim truly is not throwing as much material at you as I can. Other questions or comments or bad things or good things, maybe good things, I don't know. Nobody's heard any good things. Yes?
The amount of reading that we have to do.
The amount of reading, okay. So that's a good, yeah, it's interesting. Nobody's ever raised that as an issue before that I'm aware of. What'd you hear?
That there was
a lot of reading. Well, well, as you'll see, let me just, that brings up something else in my mind. So let me just address that sort of indirectly, okay? So one of the most common questions I get is, Well, there's so much material. What do I have to know? Okay. It's one of the worst things you can ask a professor. All right. What do I have to know for this exam? Then it gets kind of polite. People will say, well, how do I prioritize my studying? That's a politer way of asking that same question. It's still not good, but it's a politer way of asking that question. And I have a polite answer for that. And my polite answer for that is I give you information in a sort of a hierarchical fashion. The highest level of hierarchy, that is the most important things in the class, are the things that I say. If I say it in class, you're responsible for it. Okay? Okay? Pi to 100 digits. I just said it. Now you're responsible for it. Okay? Right? But if I say it in class, you are responsible for it. Okay? The second level of hierarchy is the sort of summaries that I write, the highlights, the sort of information that I give you that I said, okay, well, this is what I remember talking about. These are things that are important. I will tell you that when I go to write my exams, I sit down with the highlights in front of me, and that's where I write my exams from. That's a secret that now only 300 of you know. Okay? And the third level of hierarchy is book. All right? The book is there not for me to give you obscure things to go and find. Okay? Oh boy, I can get them on this one. Look at this amino acid. I've never seen this one before. Right? They're going to have to read this, and I didn't tell them. Right? I'm not going to do that. Okay? The book is there to supplement that information. Maybe, this may be hard to believe, I'm not the best person to describe something to you. I can't believe it, but you know, maybe it's possible. Maybe I'm not the best person to explain something. But reading it in the book, you might learn it in a different way or in a better way. Okay? That's what the book is for. So the book is supplementing what I'm talking about here. It's not there to give you additional things that you wouldn't otherwise know. Okay? Anything else? Seriously, yeah. Good question. So if you look up here, you will see that I have posted page numbers, okay? And those roughly correspond to the lectures that I'm giving, okay? There are problems, and there are no problems in this class that you will turn in. The problems are there because I think they would be useful for you to know how to do, okay? The TAs will be going through many of these problems in recitation. I would encourage you, if you want to be proactive and if you want to take charge of your education, that you try to work the problems before the TAs do. Okay? The TAs can provide a good way of checking that for you. I also have videos online of me working problems where you can take a look at those and understand those, hopefully. Okay? I frequently get asked, can I use a different edition of the textbook? Do I need a textbook? Do I have a textbook on the library? Textbook prices are absolutely outrageous in my opinion. I would love, if I could ever get any time, to put together an open source textbook where we could download the damn thing for free and everybody had that. I would love to do that. Okay, I think it'd be a very good cause, and I think I would have unanimity among students in terms of doing that because textbook prices are outrageous. If you have a different edition of the textbook, you'll see some slight changes in page number. I don't think you'll see a large difference, to be honest with you. I think you can probably get by with that. Don't quote me on that. I can't tell you in every case it's going to work for you, but I think that probably would work. If you have a textbook by a different author that is at about the same level, again, textbooks are supplementary material. So you're looking from another source at text material, that'll work. I only have one extra textbook. The TA took it over to the library this morning, and it should be in the library. It takes them about a week or so to get it posted. When it's posted, I'll let you know, and you can check that out for 2 hours at a time. And I know that's for 300 people, but that's all I've got. I don't have any other textbook to give you, unfortunately, except my own, and I have to have that for staying on top of things when people ask me questions. Okay? I will tell you there are some people who get by without a textbook. Okay? I won't advocate that, but some people do that. So there's a lot of, it depends on how you access your information. There's a lot of stuff that's online that you can find out there. So use whatever you need to access that. If you can avoid high textbook prices, I'm very much in favor of it. Okay? Okay, what else? This is good. I'm normally sweaty in a lecture, but it's even worse today. You guys probably, I need to stand back a little here. That's why they put that table here, I think, today. Just make sure I kept sufficient distance from the front row. You guys ready to dig in? If you just stop asking questions, I'm just going to start giving you stuff you're going to be responsible for. Oh, yeah, I got a question. Nobody's got a question.
Hobbies.
What are my hobbies? feel like I'm on Oprah or something. Writing. I like to write. I love to write. And I hope to share some of that with you this term, actually. So we'll see how that goes. What are your hobbies? Fishing. You know, I haven't fished in probably 50 years, which tells you how old I am, I guess. But it's been a long time. I was a kid last time I fished. Yeah. I fish for answers sometimes. You guys have probably done that on exams, too. You like to fish. Was that what you meant when you said fishing?
You haven't said anything about your songs.
I don't know anything about songs. Do you have something in mind?
No.
I don't know. I'm not sure what she's talking about. That's the funniest thing I've said all day. Yes, sir.
Can we call you Captain?
Oh, actually, you can call me anything you want to. I do prefer to be called Kevin. So please do not call me Dr. Ahern, okay? You can call me Captain. If you call me Captain, you will get 1/8 of a point of extra credit. So call me Captain, okay? But not for each time you do it. You only get, that's only redeemable once. 1/8 of a point. It might push you over that limit, but you need to make sure I know, who you are. If you tattooed your name on your head, that would be really easy. What's that? Oh, okay. So then there you go. That's good. So, Captain. I've never been called Captain before. A lot of students are very uncomfortable calling me Kevin. They prefer to call me Dr. Ahern. And if you're really, really uncomfortable with that, I won't object. But I really prefer to be called Kevin because I think doctor puts a barrier between me and you. And I don't like having that barrier. I seriously considered changing my last name to something that people would not want to call me, like Shithead, okay? Well, Dr. Shithead, I'll call you Kevin. But it's so hard getting the bank to change all your checks and everything. And when you sign your name, nobody believes you're Dr. Shithead, You guys would, though, probably. I don't know. Okay, well, it looks like we're running out of energy. You guys are thirsty for biochemistry, so perhaps I should dig into biochemistry. Are there any other questions? Yes, back in the back. Between 450 and 550, yes. The students who are in 550 are held to a higher standard, which means that their exams are graded more closely. The exam grades are set by the students in the 450 class. After those grades are set, then the 550 students' grades are assigned on top of that. So the 450 sets the curve. The 550 students do not affect the curve, so undergrads are not affected by the grad student grading. What does it mean to be held to a higher standard? Okay? 0 spelling errors on graduate student exams. That's 1. Okay? 0 spelling errors. Undergrads, I rarely allow much, but I will allow more leeway than I will allow on graduate student exams. I think by the time you're a graduate student, you should know how to spell. To be honest, I think by the time you're taking a 400 level class, you should know how to spell too. Okay? But grad students are held to that 1 higher standard there. The exams are scrutinized more carefully by me, and so I make sure that they're actually responding at that higher level. Does that answer your question? Yeah. Are you a graduate student? Boy, now you got to spell. So, okay. Other questions? Yeah.
Do you text?
Do I text? Sure. Oh wow, man. I don't tweet. I refuse to tweet. Okay. I personally think it's going to be one of the stupidest inventions of mankind. But all right. Are we set? Everybody ready to dive in? Okay. Let's dive in then. So what I want to do is just say a few general things. And this screen, it's really annoying, is not showing me anything, so I have to go up here. Okay. I post all the outlines and, as I said, all the figures and so forth that are there. I think you'll find it to your advantage to print the figures before you come here instead of trying to copy them down as I'm writing. That's one of the reasons I give them to you in advance. You can pull them down as PowerPoints, as PDFs. You can print them out. And then all you have to do is sort of scribble on them as I'm talking so that you can sort of stay on top of things with that. Okay? So that gives you an advantage in terms of taking notes. So please do that. Well, where do we start? Biochemistry. When I say biochemistry, most people have at least a reasonable notion of what it is besides the factor. Okay? Biochemistry is the chemistry of life. That was the sort of origins of the name and of the subject itself. The origins of life, or the chemistry of life. We think of the chemistry of living cells. And I say this in a bit hesitatingly because when we say something like that, that is the chemistry of living cells, we sort of imply that there's a different chemistry that happens in living cells than happens in the rest of the universe. And there's not. The rules are exactly the same. There's no difference for the rules in a cell compared to the rules outside of a cell. The environment of a cell is different. Cells are full of enzymes. We don't see enzymes out in the rest of the world. We see high concentrations of metabolites that we don't see out in the rest of the world. But the rules of chemistry are the same. Exactly the same. They don't change. So biochemistry really isn't any different from chemistry. Or in fact, you're going to like it a lot more. But it was a joke. Okay. If I have to keep telling you guys when the jokes are, it's going to be a long term. Okay? Maybe if you just got in the habit of just sort of laughing after I sort of pause, that would be a good thing. So let's try it, okay? So that's the way it's going to be. This is very good. Okay, we're off to a good start. Okay, biochemistry. Biochemistry had its roots as that, okay? It was shown back in 1829, I believe, by a man named Wooler, that there was nothing different about the chemistry that occurs in living cells. He was able to show for the first time that you could synthesize a compound known as urea completely 100% independent of a living cell. Prior to that time, it was thought that only things that were in living cells could be made in living cells. Okay? He turned that upside down. All right. Well, today we have a tremendous, and by the way, the first chapter of almost any textbook is pretty dumb, and yours is no exception. You have a pretty good textbook, but the first chapter is a little lame. Okay? The excitement over biochemistry today comes from the fact that we understand now at the molecular level a tremendous amount of information about how cells do what they do. That comes from the fact that we have sequenced the genomes of several hundred organisms, including human beings. When we say that we sequence the genomes, we know the nucleotide sequence of every single base in the human genome with 3 billion plus base pairs. All the instructions for making everything that a cell does is contained in those 3 billion plus base pairs. Okay? We're in some cases still unraveling exactly what that means. I like to tell biochemistry majors that they've got job security for the next generation or so because unraveling what all that means is a tremendous task. But at least now we have a copy of the blueprints of living cells. Okay? That's very, very exciting. It's very exciting if you are wanting to go be a doctor. It's very exciting if you want to go be a dentist. It's exciting if you want to understand anything at the molecular level. Because DNA is the ultimate molecule. It has those instructions. If we know those instructions, we can understand what proteins the cell is making to do what it does. And as we're going to see, and you're going to hear from me ad nauseum, proteins are the workhorses of the cell. Proteins catalyze the reactions. Proteins give structural support. Proteins communicate information. Proteins provide interfaces between outside the cell and inside the cell. Proteins do all of the work of a cell. Proteins protect the DNA. Chromosomes, of course. We think of chromosomes. That's the DNA. Well, chromosomes are DNA wrapped with protein. At the most basic level, protein is protecting the DNA. Proteins are doing what cells have to do. Okay? So proteins are the workhorses of the cell. If we know the instructions for making the proteins, we ultimately know everything that the cell has at its disposal. That is a tremendous advantage. You can't go today and pick up a newspaper, something hardly anybody ever does anymore, but you can't hardly pick up a newspaper and find in that newspaper an absence of articles about medical advances. Every single newspaper, every single day, has some new medical advance that's been uncovered, and the reason is because of this availability of information about proteins. Okay? It's absolutely exciting what is happening with this. Okay? And as I said, I hope to communicate with you. I'm not just going to jump up and down and say it's exciting, but I hope to show you over this term what that excitement is. You're going to see things about proteins that I think, or at least that I hope, are going to surprise you. Okay? All right. I like this example because it says, what are the similarities in the biological world? Well, there's only three things to be concerned about. First of all, there are bacteria, there are plants, and there's Linus Pauling. Okay? The three basic groups of life. Okay? You guys probably weren't aware of that, but that's the three basic groups of life. Linus Pauling, yeah. Linus Pauling, actually, I'm making fun, was an incredible scientist. He was arguably the most important scientist of the 20th century. Okay? No small accomplishment. He's an OSU alumnus, only person ever to win 2 unshared Nobel Prizes, and did some amazing things. But what this figure actually is trying to show you is that despite the differences between a bacterium, a plant, and Linus Pauling, okay, when we look at the protein level, we frequently see almost identical structures for given proteins. That tells us the very first lesson we learn in biochemistry. Structure is essential. The structure of a protein, and we're going to spend a lot of time this week, or actually mostly next week, but talking about protein structure. Protein structure determines protein function. You disturb protein structure, you disturb protein function. These are 3 proteins with very similar functions, and notice they have very similar structures. You're going to be surprised, I think, when we get into metabolic pathways, which actually won't be until later in the term. But when we talk about metabolic pathways, many of them are essentially universal. Universal. The pathway whereby I break down glucose in my body is identical to the pathway that Arabidopsis, which is a weed, breaks down glucose inside of it, which is identical to the pathway that a bacterium uses to break down glucose inside of it. The same enzymes, the same reactions, and very, very similar structures. Okay? You might wonder, how do we have the differences between a bacterium, a plant, and Linus Pauling? Okay? And that comes from the complexity with which genes are controlled. And of course, we do have some proteins that are present in some organisms that aren't present in other organisms. Plants have genes for doing photosynthesis, for example. We have genes for communicating from one cell to another in different parts of our body using hormones. Bacteria don't have that. Okay? So there are differences, but the similarities will absolutely astonish you. Okay? They still sort of take my breath away as I study the diversity in the biological kingdoms. Okay. Why does that show up in the first chapter? It's like, we got to throw it in there somewhere. It's not a biochemistry book unless we talk about adenine, guanine, cytosine, and thymine. So here it is in the first chapter. There's 4 structures for you to memorize, okay? I was pausing. You see, if you laugh, then I may not hold you responsible for it. However, if you're just going to go, oh, God, there's another one for me to learn, well, maybe he's serious. Maybe he'll have to do this, okay? So maybe a safety valve would be, in fact, if you just kind of giggled. And I'll say, no. You do not need to know these structures, okay? You don't even need to know these names at this point. Base pairs, okay? A pairs with T, G pairs with C. Is there anybody in this room who has never heard that? Be honest. Okay, so we've covered that. Okay, moving ahead. And there's another base pair. Look at that. And DNA, in case nobody's ever heard this, replicates. Has anybody not heard of that? Okay. That's where the A pairs with G. Wait a minute. A pairs with... You get it. Okay. All right. So, DNA replicates. DNA replication, of course, is essential for everything that we know about biology because it gives rise to us. It gives rise to duplicates of our cells. We all started out as a single fertilized egg that duplicated itself trillions of times, virtually identically. Notice I said virtually identically. Replication is not perfect. No matter how much the cell works, it cannot replicate perfectly, which gives rise to diversity from generation to generation, which gave rise to the diversity of life that we see on the face of the earth. Pretty exciting. Without that, we are still bacteria. Okay, and now we move into some real things. Okay, so much for the opening chapter material. Let's talk now about the beginnings of structure. And I'm going to teach you something about protons and about hydrogen bonds that your freshman chemistry classes probably didn't teach you. Why? Because they probably decided that here's a whole bunch of stuff for you to memorize, and you went and you dutifully memorized that stuff, and you probably didn't learn it. Okay? I want you to learn it. I really want you to learn it. So I'm going to describe it to you probably in some ways different from what you've heard before. I start with the hydrogen bond. Oh, I know what a hydrogen bond is. It's between a donor hydrogen and an acceptor oxygen, and that's what a hydrogen bond is. And it is. Okay? Donors and acceptors. But people get so wound up with memorizing what's a donor and what's acceptor, they don't really understand what a hydrogen bond is and the significance of it. And as it turns out, hydrogen bonds in biochemistry are very, very important. There's no way of escaping hydrogen bonds in biochemistry, so we need to understand hydrogen bonds first. Well, how does a hydrogen bond differ from a covalent bond? Well, you probably, when you were in freshman chemistry, you memorized it. You said, well, there's an energy difference between the two. Anybody get that? Anybody get that in freshman chemistry? Is that what you got? Covalent bonds are harder to break, higher energy, right? They're harder to break. That's exactly right. They are harder to break. The bond between an H and an O in a water molecule is different than the bond between an H and an O between water molecules. The H and the O bond in a water molecule is a covalent bond. The H and the O between water molecules is a hydrogen bond. Hydrogen bonds arise as a result of unequal sharing of electrons. There's probably something else you memorized. What does that mean? It means that the oxygen atom, which is the most common atom, it's not the only one, but the most common atom involved in hydrogen bonds, holds electrons closer to it than hydrogen does. Okay? So, if I look at this guy down here, for example, here's a portion of a water molecule. Here's an oxygen on another water molecule. This oxygen over here is holding electrons closer to it than hydrogen is, which means that this oxygen is partially negative. It's not fully negative because that would be an ionic bond. It's partially negative. So we can think of it as a 1/3 of a negative charge if you want to think of it that way. We're going to put a number on it. Okay? It's partially negative. That makes the hydrogen partially positive. However, this hydrogen is attached to it. They are sharing electrons. Not equally, but they're sharing electrons. This hydrogen is physically attached to it. This hydrogen is not physically attached to this oxygen, which also happens to be partially negatively charged. Well, if the oxygen is partially negatively charged, it's taking electrons away from the hydrogen. That makes the hydrogen partially positively charged. Now you see the basis of a hydrogen bond. It's simply attraction. Okay? The partially negative oxygen attracts the partially positive hydrogen, and they now associate with each other. That has tremendous importance. Without this bond right here, we don't have life on Earth. Okay? Without that bond right there, we don't have life on Earth. Those attractive forces make water a liquid at room temperature. Water is liquid at room temperature because it has hydrogen bonds. Water has a molecular weight of 18. Methane has a molecular weight of 16. They're very, very similar. Carbon dioxide has a molecular weight of 44. Methane and carbon dioxide are both gases at room temperature. Why? Even though they've got similar molecular weights, the answer is neither carbon dioxide nor methane have hydrogen bonds. Hydrogen bonds are attractive forces that are holding molecules close together and allowing water to be a liquid at room temperature. Okay? Now, how do we break hydrogen bonds? Well, we break hydrogen bonds with heat. We can't break covalent bonds with heat unless we go to several hundred degrees Fahrenheit. Hydrogen bonds we can break with heat by heating it up to the temperature of boiling water. What we're doing when water boils is we're breaking hydrogen bonds. The waters are escaping from each other and going off into the atmosphere. Okay? Make sense? I don't mind questions. You can always ask questions in here. You can also raise your hand and say, hey, Kevin, slow down. You're going too fast again. Okay, so hydrogen bonds are essential. Well, okay, so life relies on liquid water. All right? La-di-da, how many times have I heard that? Right? Hydrogen bonds do much more than make water be liquid at room temperature. Hydrogen bonds are one of the fundamental forces that give proteins the structure that proteins have. Why do we have such a weak force giving proteins their structure? Okay? It's only one of several. Proteins have covalent bonds as well. But hydrogen bonds contribute to giving proteins their structure. For DNA, it turns out that when we look at those base pairs that are between the strands, the base pairs are held together by hydrogen bonds. The only things holding all of your double helices together are hydrogen bonds. Wow. Makes you feel kind of unstable, right? Well, it turns out that collectively they add. Collectively they add. So over the course of a chromosome of a few 100 million base pairs, that's a reasonably strong force that holds those strands together. But over the short range of a few base pairs, where a DNA polymerase has to get in and start replicating things, it's relatively easy to peel apart those few bases. Hydrogen bonds give us the beauty of strength in numbers, weakness in small numbers. Because of that, it's relatively easy for the cell to pull apart strands when it needs to do something. If the cell had to break covalent bonds to separate the strands of DNA, we would never have replicated a single DNA. That's the power of hydrogen bonds. Okay? All right. So, as you can see, hydrogen bonds occur between things that are something besides oxygen and hydrogen. Okay? Here's an NH. Nitrogen has a greater electronegativity than does hydrogen, so it holds electrons more closely. Partial negative charge, partial positive charge. This type of bond happens frequently in proteins. This is what an amino acid has hanging off of it. This is an oxygen that is also found in amino acids. So now we see how the hydrogen bonds within proteins can form. All kinds of possibilities of these right here. Okay. And there's some dimensions, okay? If you really want to get bored, you can memorize those dimensions, all right? But you don't need to do that. You should know the following about hydrogen bonds with respect to dimensions. They really only work over a fairly short distance. You start pulling things too far apart, the hydrogen bonds do not exist as a force or as a significant force. Okay? You got to have them pretty close together. Now, you might think the closer you get them together, the better. And the answer to that is no, that's not right either. Because if you try to push two atoms too close together, you run into problems. The problems are illustrated in this figure right here. The van der Waals contact distance. All this says is that basically there's an ideal distance of attraction between two atoms. An ideal distance of attraction. That's all that this says. It says if you are at that ideal distance of attraction, that's where you're going to see atoms separated by each other, because this is where they want to be. If you try to pull them too far apart, that attraction gets less. It's getting closer to zero. The further you pull them apart. The closer you try to push them together, look at that repulsive force. The repulsive force beyond a certain distance goes up to the 12th power as you try to squeeze them together. Well, why do we want to squeeze them together? We don't want to squeeze them together, but what we're going to see when we talk about protein structure is that proteins fold, and they fold in specific ways so as to avoid getting too close together. Because if two atoms start getting too close together, the repulsive force will push them away. That repulsive force can be very, very strong. Okay? So distance really matters. Because distance matters, we start beginning to see the glimmers about why shape matters. As we change distances, we're going to change shapes. Shape is very important. And we can change shapes. Okay? You betcha we can change shapes. I said we can denature, or we can, we can break hydrogen bonds with heat. That's why you cook your food. You cook your food, so you break the hydrogen bonds in the proteins of the bacteria that would otherwise give you food poisoning. Right? And once you've denatured their proteins, they don't come back together very nicely. You kill the bacteria. That's how you kill them. Because once you've pulled those proteins apart, for the most part, they won't come back together. We'll talk a little bit about that later. You've destroyed the shape doesn't come together. Welcome. How you doing? People are so interested in this class, they just kind of come walking in. It's kind of cool. They're so embarrassed. All right. Let's see. Hydrophobic. All right. Another force that we'll talk about, and I'm just going to briefly bring it up here, is a force called the hydrophobic force. Now, you know what the hydrophobic force is, but you probably never thought about it a lot. All right? The hydrophobic force explains why water and oil don't mix. And you say, well, I know water and oil don't mix. And if I were to ask you at the energetic level why water and oil don't mix, and you're not a chemistry major, I'll wager that nobody in here could tell me. Okay? I'll wager that nobody in here could tell me. Should I tell you? No, we're running out of time. We'll save that for next time. See you guys then, and happy biochemistry. Okay.
Hello, Kevin.
Hey, how you doing, John? Hey, this thing is acting up, so we've had some problems. The screen blacked out once about halfway through the lecture, so
Okay.
I had to be looking back the whole time to do it.
Okay. All right. Well, I guess they.
That's why I shut it down here, so that it would come,
right,
back on.
Do I need to?
You'll probably have to go through all that business.
Okay.
Here's
All
your
right.
microphone.
Thank
We're
you.
back for another term, and the microphone is lacking a clip, so I had to clip it using my own clip here. Yeah, welcome to the first day of class, right?
Lovely.
Yes.
Hi.
Just a second here. So there you are. You may need to tie it onto your shirt or something. Yeah, I can't give you my clip, unfortunately. And I think that should do you. I had a little bit of trouble with the projector at first, but they got it going. So.
Excuse me.
Okay. Hi.
I'm Danielle. I emailed you last week about missing class.
Okay. Missing this class? Okay.
Missing this class on Friday because I'll be at a conference.
Okay.
So I won't be able to attend. I'm signed up for Thursday recitation.
Okay.
Is that going to...
Are you miss Thursday recitation?
I will miss Thursday recitation.
Well, missing one's not going to kill you.
Okay.
Okay?
All right.
What is the book about?
It's a blue book, but I don't have one here, but it's a blue book. It's by Berg, Tomsko, and Stryer.
Okay.
It's got a, what's it got on the front of it? Like a DNA molecule, I think.
Okay.
Yeah. It should be available in the library. If you want to come by my office, I'll show you one.
No, it's okay. I'm going to go to the bookstore. I just wanted to make sure I got it.
Yep. Hopefully they still have some used ones.
Okay.
Okay?
Thank you.
Good luck. Take care. Hi, how you doing? The grad student, how you doing?
Hi.
I picked on you there, didn't I?
Yeah, no, not at all. It's fine.
Yeah. Sit back this way. Yeah. What's up?
Well, I just wanted to ask you about registering for the class.
Oh, okay. You have trouble getting in?
Yeah.
Oh, okay.
There appears to be spots open in the
Sure.
lecture part. So the recitations are closed.
Okay. So that's fine. What you need to do is go to room 2011
Okay.
and talk to Barbara Hansen and tell her what you told me, and she'll give you an override to get into the class.
2011 Barbara Hansen.
ALS 2011.
ALS.
Okay. I'm headed back there in a minute if you want to wait, but I got to unload all this first. So.
Okay.
But if you go back now.
Well, I'll head that way.
Okay.
Thank you.
Yeah. Any problems? Let me know.
Quick question for you, Lloyd.
Yes, sir.
There's videos on iTunes.
Yep.
Are those from last year? Are those going to be